Assalamualaikum and a very good evening. So today we will do experiment 4.2 digestion of starch. This is using iodine test. So test tube A1, it contains glucose, and after added with iodine, it changed from colorless into yellow. Thus, it does not contain any starch because yellow is the color of iodine. For test tube B1, it contains of starch and added with iodine. It changed from chalky into blue-black. It shows that there is a presence of starch. For test tube D1, it contains starch and untreated amylase. It changed from chalky into blue-black. So this means there is a presence of starch also. And for test tube E1, it contains starch and acidified amylase. It changed from chalky into blue-black. So that means there is a presence of starch.
Hi guys, so today I'm going to show to you the result of the digestion of starch. Okay, to detect the presence of the reducing sugar, so I use the Benedict reagent. Okay, so the first test tube, it contains only glucose, and the color change from blue to orange. So it means that there is the presence of the reducing sugar in this test tube. So the next test tube is B2. Okay, in B2, it contains only starch. Okay, so there is no change in this, no color change. Okay, so it means that there is no presence of reducing sugar in this test tube. Okay, so the third test tube is test tube D2. Okay, for D2, it contains starch and untreated amylase. Okay, so there is change, the color change from blue to yellow. Okay, so it means that there is the presence of reducing sugar in this test tube. Last test tube, okay, E2. Okay, for E2, it contains starch and also acidified amylase. Okay, but actually the result should be no change, but we had made some mistakes. So later we will explain why the result should not be like this. So I think that's all for this one. Thank you. Hi guys, so I'm going to explain to you about the mistake that we have been made in our last test tube, which is E2. Okay, there are some trouble during we handle the experiment. So actually the result should be no change. Okay, but we get the result against what we expected. This is because the concentration of HCl is not enough to reduce the enzymatic reaction in the solution. Okay, so that's why it changed, and it can't reduce the enzymatic reaction in the test tube, in the solution. So I think that's all. Thank you.
There are three tubes that have color changes, which is test tube A, test tube B, and test tube D. These tubes have color changes because they contain all fatty acid, and the test tube C doesn't have any color changes because it doesn't contain fatty acid. That's all for us. Thank you.